Here we go. The last topic that's going to be on your cells test. We're going to be discussing about cell transport, how these substances actually can get inside and out of your cells through the cell membrane. What molecules can cross directly through the cell membrane? The cell membrane, remember, is a double layer of phospholipids. It has polar heads and has nonpolar tails, meaning that water cannot actually directly pass the membrane. There's only one type of molecule that can really go straight through the membrane without a door, and that is fats and oils. Because the interior is made of fat, small, tiny, non-polar molecules can actually squeeze themselves through the head and can travel all the way through the other side. As you can see, lipids can go directly through the membrane without any issues, while other things cannot. So the question is, what about the other stuff? How do we get the other materials needed for your cells actually through the cell membrane if they cannot directly walk through the wall? You can see sugar, amino acids, water, all of them cannot directly pass through the membrane. We actually make doors to the membrane. What are these doors made from that different things can get through? They're actually made out of proteins. So proteins form the channels. This is actually what it looks like, which you'll never probably see that diagram again. So for our purposes, we can just think of them as little tunnels that different substances can get through. So most substances actually need to go through protein channels to get inside and outside of your cells. Each channel is highly specific. We have a channel that's made just for water, a channel that's just made for amino acids, a channel that's just made for waste products. It's a highly specific. If you are not water, then you cannot pass through any other door but the water door. Here we have the sugar channel, so sugar can pass through, an amino acid channel, a water channel known as an aquaporin, salt channel, waste channel. Each individual substance needs its own personal door, personal pathway into and out of your cells. How do you actually build a semipermeable membrane? We have the bilipid membrane. We have these channels that are made of proteins. These proteins both like water and like lipids, so they're able to completely integrate into the membrane and form those actual tunnels that different substances can get through. Once again, the proteins act as these doors in the membrane. They're going to allow the specific molecule to actually get through the cell membrane. You can see a picture. The substance is going to travel through the doorway that it's been assigned to get into or to get out of your cell. Also notice which way the things are moving. They're moving from an area where there's lots of this substance to an area where there's not a lot of this substance, which we'll come back to in a second. Why do things actually move if you give them the doorway? If you have 50 students shoved in my classroom tomorrow, and I open the doorway, I think we all know what's going to happen. You're all going to go rushing through the door and out. The question is why? Why are they actually going to start moving? Well, let's look at the picture. Up top, we have 1, 2, 3, 4, 5, 6 molecules of this material outside of the cell. And we only have 2 on the inside. So just like how if we had 50 students shoved in my classroom up here and nobody in the hallway, the second I open that door, you're all going to rush out from an area of high concentration up here to an area of low concentration down here. So just like how you fall down the stairs due to gravity, you're going to move from an area of high concentration to an area of low concentration. This movement from high concentration to low concentration is known as diffusion. So diffusion is when molecules move from a high area of concentration to a low area. There is known as moving down the concentration gradient. When you have a concentration gradient, that just means you have a different amount of substance on each side of the membrane. One side you have 10 molecules, the other side you have 2. That is a concentration gradient. And during diffusion, you always flow down the concentration gradient from high concentration to low, going downriver, just relaxing, going down the concentration gradient. Here we can see we have a concentration gradient, lots of concentration that side, zero concentration that side. You open up the pathway, open up the little pores, and we can see over time that eventually it's going to equal out. One thing I would also like to mention in this picture, students have this misconception that once we have an equal number on each side, just movement stops. It's not going to stop. As long as something has a temperature, these molecules are still banging around. All it means with equilibrium is that for every one guy that goes this way, one guy is going that way. So there's always equal number, but they're constantly exchanging sides. Diffusion is known as passive transport. If something is passive, like hopefully you're not doing while watching this video, but after you get home from school, you're probably very passive once you lay on that couch. So passive means that you have no energy needed. So passive transport means that the cell doesn't actually have to do any work to get the materials to move. So here we have diffusion of water, we have normal diffusion, so diffusion is a passive transport. No energy is required from the cell. When you move directly through the membrane, that is known as simple diffusion. So these tiny non-polar molecules, these tiny fat molecules, go through simple diffusion, can go directly from high to low, through the wall, through the membrane, no door needed. When you need a channel, when you need a helper, when you need a doorway to actually get through the cell membrane, that's called facilitated diffusion. Facilitated or facilitators are helpers. So teachers are facilitators. We're helping you gain the knowledge. So facilitated diffusion means you're still going from high to low, you're still not requiring energy, but you do need that help of a door. You need an opening to actually pass through. So this is how sugar and amino acids are going to get into your cells. Another comparison, simple, we can see it directly through the membrane, while facilitated has to go through that protein channel. But once again, both are still from high to low, and both do not require energy from the cell. They're passive processes. The next thing I like to discuss is how does water actually move across your cell. Water is extremely important for life, obviously, and because it's so special and so important, we call it a different name. We call it osmosis. Osmosis is exactly the same thing as diffusion. It is literally defined as the diffusion of water from where there's lots of water to where there's not a lot of water. So osmosis is actually water molecules moving. Diffusion is any other type of molecule moving. Here we have an example which is quite complicated, which I'll really explain in class tomorrow. We can see here that we have so much water on this side, so much water on this side. After time, the water is actually going to move from left to right. And it's actually going to unbalance the levels. So we can see originally we had an equal water level on each side. Over time, it's actually going to move down here and up here. And the question is why? Well, if we look at the actual picture, we notice this side has way more dissolved particles in it than this side. Therefore, we say that this side actually has more water molecules. And water is going to move from an area where there's lots of water to an area where there's not a lot of water. So the more stuff you have dissolved in your water, the less free water molecules there are. Therefore, the water molecules are going to move from where there's lots of them over here to where there's actually not a lot of them on this side. And once again, I'll go over this in much more detail in class to make sure you understand this idea of water's moving. It's actually individual free molecules that are moving from a high concentration to a low concentration. There's 3 different types of conditions we talk about with osmosis. We have a type where the cells actually can burst. Animal cells burst, while plant cells love it. It's their normal condition. We have another condition where animal cells love it. They're normal, while this is actually the condition that causes plants to go droopy. And then we have a condition where water is actually sucked out of the cells, and plants also get sucked out, but it's much worse for animals than plants because of the cell membrane. This is what happens when you put your cells in fresh water. Your cells actually don't like it, and they go boom. If we put your cells in salt water, those salt water sucks out the water, and your cells shrivel up. And when you put them in a balanced solution, this is what we give you when we give you an IV fluids. When we give you an injection, it's actually going to keep your blood cells nice and plump. The first type of condition, which is the freshwater condition, is known as a hypotonic solution. Hypotonic. Hypotonic means that there's more water outside the cell than inside the cell. Therefore, if there's more water outside than inside, water is going to flow into your cell. So it's going to go from high concentration outside to low concentration inside the cell. This causes the cell to gain water, and if you're an animal cell, you're going to go boom. If you're a plant cell, you have this wonderful cell membrane that stops you from going boom. It's like putting a balloon in a plastic box. You can blow it up all you want, but if the plastic box is small enough, the balloon will not pop. The next condition we have is a saltwater condition, known as, or any type of solution, which is a hypertonic solution. Hyper is the exact opposite of hypo. So hypertonic solutions is when there's not a lot of water on the outside. Because you have a lot of salt, you have a lot of dissolved particles, there's not a lot of water outside the cell, so water is going to actively leave the cell and is going to cause your cell to shrivel up and die. Same thing with plants, they can survive it better because of the cell wall, but even they do not like to have salt water. So the cell loses water in a hypertonic solution. Hypo, it gains and goes boom. Hyper, it shrivels up. That's why you should not drink salt water if you're ever stuck at sea, because you're just going to cause your body to dehydrate faster. The last condition is known as isotonic. Iso means balance, so isotonic means a balanced condition. Therefore, there's no difference in concentration between the outside and the inside. There's a certain amount of water on the outside, there's a certain amount of water on the inside. So water is going to exchange at an equal rate. It's not going to not exchange. Just for every one water molecule that goes in, one's going to leave, which for animals is perfect. It's Goldilocks, it's balanced, it's just right. It's going to be the normal condition. While in plants, it's actually not so great. It's going to cause them to wilt. So when you give them an isotonic solution, they're actually going to wilt and become flaccid. So isotonic means that the cell is in equilibrium. It's not going to gain water, it's not going to lose water. It's just going to stay perfectly balanced. The last type of transport I want to talk about is active transport. Active transport is the exact opposite of passive. So we said passive is going down the concentration gradient from high concentration to low concentration. Active transport is now we're going uphill. Sometimes you need more of a substance than you actually can just rely on passive transport. So you're actually going against the grain. You're swimming upriver, you're walking up the stairs. So it's known as active because you're actually using energy. You're going from a low concentration up to a high concentration, and the cell actually has to use energy to do this. Usually you have protein pumps that are going to do this. They're going to use ATP, and they're going to pump the molecules. Notice there's not a lot of amino acids here, but we still need more and more amino acids, so we're going to pump them into our cells using energy in the form of ATP. The last thing I want to discuss is just a quick review of the types of transports we saw. We had the two types of passive transport. We had the first type where the materials move from a high concentration to a low concentration directly through the membrane, which is simple diffusion. We have the second type where we're going from high concentration to a low concentration through a channel, which is facilitated diffusion. And the last type we just discussed is when you're going from a low concentration to a high concentration using ATP and a protein, and that is active transport. So we have two types of passive and only one type of active. Passive, high to low, active, low to high.